This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome to this exploration of Hox genes, the master architects of the animal kingdom. In this easy-to-understand journey, we'll unravel the secrets of these remarkable genes, revealing their profound influence on shaping the diverse forms of life on Earth, from the humble fruit fly to ourselves. Prepare to be amazed as we delve into the intricate world of Hox genes, discovering how they orchestrate the development of our bodies, how their misregulation can lead to surprising transformations and diseases, and how scientists are harnessing their power to revolutionize medicine. Over the course of this exploration, we'll uncover the fundamental principles of Hox gene function, exploring their role as master regulators of development. We'll learn how these genes, like skilled conductors, orchestrate the intricate symphony of development, ensuring that the right structures emerge in the right places at the right times. We'll delve into the concept of the Hox code, the combinatorial expression of Hox genes that determines the identity of different body regions, and we'll see how this code, while remarkably conserved across diverse species, also allows for the incredible variety of animal forms we see today. Our journey will take us through the evolutionary history of Hox genes, tracing their ancient origins and their remarkable conservation across vast spans of time. We'll explore how these genes, while maintaining their core function, have also diversified and adapted to the unique needs of different organisms, contributing to the breathtaking array of body plans found in the animal kingdom. We'll also investigate the darker side of Hox genes, examining how their misregulation can disrupt the delicate balance of development, leading to surprising transformations and diseases. We'll explore the link between Hox genes and cancer, revealing how these master architects can sometimes go rogue, their precise instructions becoming scrambled and leading to uncontrolled growth and the formation of tumors. Finally, we'll look towards the future, exploring the exciting frontiers of Hox gene manipulation and its potential to revolutionize medicine. We'll delve into the promise of regenerative medicine, where Hox genes could be harnessed to repair damaged tissues, regenerate spinal cords, and even regrow lost limbs. We'll also examine the challenges and ethical considerations that accompany these powerful new technologies, ensuring that their application is both safe and responsible. By the end of this exploration, you'll have a deeper understanding of the fundamental principles of development, the intricate mechanisms that shape our bodies, and the remarkable potential of Hox genes to transform medicine. Get ready to embark on a fascinating journey into the world of Hox genes, where we'll uncover the secrets of life's blueprint and explore the future of form. Chapter 1. From Flies to Humans: The Remarkable Journey of Hox Gene Evolution. Imagine a play unfolding on the grand stage of life, where genes act as directors, choreographing the intricate dance of development. Among these genetic directors, the Hox genes stand out as master regulators, wielding immense power over the body's architectural blueprint. Their story begins with a seemingly humble character, the fruit fly, Drosophila melanogaster. In the early 20th century, scientists studying these tiny creatures stumbled upon bizarre transformations that would revolutionize our understanding of how bodies are built. Flies with legs sprouting from their heads, where antennae should have been, or flies boasting a majestic 4 wings instead of the usual 2. These were the startling results of mutations in Hox genes. These homeotic transformations, as they are called, provided the first glimpse into the hidden world of Hox genes. The antennapedia mutation, for example, caused the fly's antennae to be replaced by legs, a dramatic shift in appendage identity. The bithorax mutation, on the other hand, resulted in the transformation of halteres, the small balancing organs on the fly's third thoracic segment, into a second pair of wings. These mutations, like mishaps in a play's script, revealed that Hox genes were responsible for assigning the correct roles to different body segments. A pioneer in this field, Ed Lewis, meticulously mapped these mutations and their corresponding genes, laying the foundation for our understanding of Hox gene function. Hox genes, it turns out, are just one part of a larger family of genes known as homeobox genes. These genes encode transcription factors, the protein messengers that bind to DNA and control the activity of other genes. Like a director cueing the actors, transcription factors dictate which genes are switched on and off, ultimately shaping the characteristics of a cell and its contribution to the body's overall design. Within each Hox gene lies a highly conserved DNA sequence called the homeobox, a molecular signature that gives these genes their name. This sequence encodes a corresponding protein domain, the homeodomain, which acts as the transcription factor's DNA binding interface, allowing it to latch onto specific regulatory regions of its target genes. The influence of Hox genes extends far beyond the placement of appendages in flies. In insects, they determine the types of appendages that form on each segment, legs, antennae, wings, or mouthparts. In vertebrates like us, Hox genes orchestrate the development of the vertebral column, specifying the types and shapes of the vertebrae that will form along the spine. Even in the seemingly simple worm, Hox genes play a crucial role in establishing the body's segmented structure. The story of the Drosophila head provides a compelling example of Hox gene precision. Genes like labial, proboscipedia, and deformed sculpt the intricate architecture of the fly's mouthparts. Sex combs reduced plays a role in head and thorax development, while antennapedia, as its name suggests, is involved in antennal formation. Mutations in these genes can lead to a range of developmental errors. For instance, a labial mutation can disrupt head involution, a critical process where the fly's head structures, initially developing on the outside of the body, are internalized. This can lead to missing or malformed mouthparts and salivary glands. A deformed mutation can cause similar head defects or even transform head segments into thoracic segments. A dominant antennapedia mutation, famously, can result in legs growing from the fly's head in place of antennae. The expression of Hox genes follows a remarkable pattern known as collinearity. Spatial collinearity refers to the correspondence between the order of Hox genes on the chromosome and the order of their expression along the head-to-tail axis. Temporal collinearity adds another layer of coordination. Hox genes at the 3' end of the cluster are expressed earlier in development than their 5' counterparts. This means that the position of a Hox gene in the cluster reflects both the timing and location of its activity. This principle can be observed in the expression of Hox genes 7, 8, and 9 in arthropods, where mutations can shift their expression domains by up to 3 segments, altering segment identity. The evolutionary journey of Hox genes is a testament to their enduring importance. These genes are present across a vast array of animals, from bilaterians, animals with a clear head-to-tail axis, to cnidarians, like sea anemones. Their presence in such diverse groups indicates an ancient origin, predating the Cambrian explosion over 550 million years ago. In many animals, Hox genes are organized in clusters, although chromosomal rearrangements have sometimes disrupted this arrangement. The remarkable similarity of Hox gene sequences between different species, often greater than the similarity within a species, points to an early evolutionary history of tandem duplication and divergence. A prototypic Hox gene cluster, containing at least 7 different Hox genes, is thought to have existed in the common ancestor of all bilaterian animals. The functional conservation of these genes is astounding. The fact that a fly can largely function with a chicken Hox protein replacing its own highlights the deep similarities in their molecular mechanisms. Drosophila, with its well-characterized genetics and short lifespan, has become a powerful model for understanding Hox gene function. Its 8 Hox genes, clustered into 2 complexes, the antennapedia complex and the bithorax complex, provide a simplified system for studying the intricate logic of body plan development. The principles uncovered in Drosophila apply broadly to all bilaterian organisms, including humans. The labial gene, expressed in the head, is essential for head involution. Proboscipedia shapes the mouthparts, while deformed patterns the maxillary and mandibular segments. Sex combs reduced contributes to head and thorax development, and antennapedia specifies the identity of the second thoracic segment, promoting leg and wing formation. The ultrabithorax gene patterns the third thoracic segment, repressing wing development to form halteres. Abdominal A specifies the identity of most abdominal segments and represses limb formation, while abdominal B is involved in the development of the posterior abdomen. Mutations in these genes can lead to a range of developmental defects, mirroring the transformations observed in the early Drosophila mutants that first brought Hox genes to light. The story of Hox genes is a story of remarkable conservation and evolutionary ingenuity. These genetic directors, with their ancient origins and powerful influence, have shaped the body plans of countless creatures, from flies to humans, uniting us in a shared genetic heritage. Their intricate dance of regulation, choreographed over millions of years, continues to unfold in every developing embryo, a testament to the elegance and power of nature's design. Chapter 2. The Hox Code, Orchestrating the Body's Blueprint. Previous chapter introduced us to the Hox genes, master regulators of development that dictate the body's architectural plan. But the true marvel of these genes lies not just in their individual roles, but in their intricate coordination, their harmonious performance as a genetic orchestra. This chapter delves deeper into the Hox Code, exploring the elegant mechanisms that ensure the right structures form in the right places at the right times. The Hox genes are not scattered randomly throughout the genome. Instead, they are arranged in clusters, like sections of an orchestra, with the position of each gene in the cluster reflecting its role in the developmental symphony. This arrangement is the foundation of spatial collinearity, where the order of Hox genes on the chromosome corresponds to the order of their activity along the head-to-tail axis of the developing embryo. For instance, in the fruit fly, Drosophila, the labial gene, located at the 3' end of the antennapedia complex, is expressed in the most anterior part of the embryo, while abdominal B, at the 5' end of the bithorax complex, is expressed at the posterior end. This spatial arrangement ensures that the head forms at one end and the abdomen at the other. But the Hox Code is not just about spatial organization. It also incorporates a temporal dimension, known as temporal collinearity. This principle dictates that the timing of Hox gene activation also corresponds to their position in the cluster. Genes at the 3' end are expressed earlier in development than those at the 5' end. This temporal coordination is crucial for the orchestrated progression of development. In vertebrates, for example, Hox genes are first activated during gastrulation, the process where the embryo forms its 3 primary germ layers. The 3' Hox genes are expressed first in the anterior regions, setting the stage for the development of head structures, while the 5' genes are activated later in the more posterior regions, guiding the formation of trunk and tail structures. This elegant spatio-temporal control is akin to a play director orchestrating a complex performance. The Hox genes, as directors, do not perform the actions themselves. Instead, they cue the actors' downstream genes involved in the actual construction of body structures. In insects, Hox genes specify the types of appendages that form on each segment, legs, antennae, or wings in fruit flies, but they don't build the segments themselves. The antennapedia gene, for instance, dictates that the second thoracic segment will develop legs and wings, but other genes are responsible for the actual growth and differentiation of these structures. This division of labor is essential for the precise and coordinated development of complex body plans. The play director analogy extends further when considering the different developmental strategies of insect larvae. In larvae with gradual metamorphosis, like grasshoppers, Hox genes are expressed in larval tissues that will be carried over into adulthood, ensuring the continuity of body plan. However, in larvae with complete metamorphosis, like butterflies, Hox genes are primarily active in the juvenile rudiments, the nascent structures that will give rise to the adult form, while being largely absent in the transient larval tissues. This difference reflects the dramatic transformation that occurs during complete metamorphosis, where the larval body is essentially broken down and rebuilt into the adult form. Hox genes act as molecular addresses, providing positional information along the body's 8p axis. They determine the identity of each segment, ensuring that the correct structures develop in the correct location. In Drosophila, the labial gene, located at the 3' end of the antennapedia complex, is the most anteriorly expressed Hox gene. It is crucial for head involution, the process where the embryonic head structures are internalized. Mutations in labial can disrupt this process, leading to severe head defects, including missing or malformed mouthparts and salivary glands. Other Hox genes, like proboscipedia, deformed, sexcombs reduced, and antennapedia, further refine the head and thorax segments, specifying the development of various structures. Moving further along the Drosophila body, we encounter the bithorax complex, which governs the development of the thorax and abdomen. Ultrabathorax is key to patterning the third thoracic segment, where it represses wing development and promotes the formation of halteres. Mutations in ultrabathorax can lead to the iconic four-winged fly, where the halteres are transformed into a second pair of wings. Abdominal A specifies the identity of most abdominal segments and plays a critical role in repressing limb formation in the abdomen. Abdominal B is involved in the development of the posterior abdomen and influences cuticle patterns. These genes, acting in concert, orchestrate the intricate development of the fly's segmented body. The arrangement and function of Hox genes, while remarkably conserved, also exhibit variations across different animal groups. Arthropods, like insects and crustaceans, typically have a single Hox cluster, while vertebrates have four: Hox A, Hox B, Hox C, and Hox D. This expansion of Hox clusters in vertebrates likely contributed to the increased complexity of their body plans. The four clusters provide a greater repertoire of Hox genes, allowing for more refined control of development. The combinatorial expression of Hox genes from these four clusters creates a Hox code, where different combinations of Hox proteins specify the identity of different regions and structures. This Hox code is exemplified in the development of the vertebral column in mice. Different vertebral elements, cervical, thoracic, lumbar, sacral, are specified by unique combinations of Hox genes. Mutations in specific Hox paralog groups can lead to transformations of one vertebral type into another, illustrating the crucial role of the Hox code in establishing vertebral identity. For example, mutations in Hox 5, Hox 6, Hox 9, Hox 10, or Hox 11 genes can cause alterations in the shapes and characteristics of specific vertebrae, demonstrating the fine-grained control exerted by the Hox code. The organization of Hox genes also reflects evolutionary history. In arthropods, the degree of overlap in Hox gene expression correlates with body plan complexity. Crustaceans, with their numerous similar segments, show substantial overlap in Hox gene expression, while insects, with more specialized and diverse segments, exhibit reduced overlap. This difference reflects the evolutionary transition from a relatively homogeneous body plan to one with greater regional specialization. The reduced overlap in insects allows for more distinct combinations of Hox genes to specify the unique identities of different segments, contributing to the remarkable diversity of insect body forms. The Hox code is a testament to the power of genomic organization and gene regulation in shaping biological form and function. From the humble fruit fly to the complex vertebrate body, Hox genes orchestrate the developmental symphony, ensuring that the right structures form in the right places at the right times. Their ancient origins and enduring influence highlight the deep connections that unite all animals in a shared evolutionary history. As we continue to unravel the intricacies of the Hox code, we gain deeper insights into the fundamental processes that have shaped the diversity of life on Earth. Chapter 3. The Architects of the Body: Unraveling the Secrets of Hox Genes. Previous chapter delved into the Hox code, the intricate spatio-temporal regulation of Hox genes that orchestrates the body's blueprint. This chapter further unravels the secrets of these master architects, exploring the molecular mechanisms by which they exert their control over development, and the profound consequences when their meticulous instructions go awry. Hox genes don't build the body directly. Instead, they act as molecular foremen, delegating tasks to the construction workers, the downstream genes responsible for the actual building process. The protein products of Hox genes are transcription factors, molecular messengers that bind to specific DNA sequences called enhancers. These enhancers act as control panels, regulating the activity of hundreds of other genes involved in building the body. A single Hox protein can be a repressor for one gene and an activator for another, demonstrating its remarkable versatility. This ability to bind DNA resides in a specific region of the Hox protein called the homeodomain. This 60 amino acid long sequence is the key to the Hox protein's regulatory power. It folds into a characteristic helix-turn-helix motif, a three-dimensional structure that fits snugly into the major groove of DNA. The third helix of this motif acts as a stabilizing anchor, ensuring a strong interaction between the Hox protein and its target DNA sequence. This precise molecular handshake allows the Hox protein to either activate or repress the expression of its target genes, orchestrating the complex processes of development. The Hox protein's regulatory influence is further refined by its collaboration with cofactors, like PBC and MACE proteins, which are themselves encoded by other homeobox genes. These cofactors can modulate the Hox protein's DNA binding affinity or its ability to interact with other components of the transcriptional machinery. This intricate network of molecular interactions underscores the complexity and precision of Hox gene regulation. One striking example of Hox gene regulation is the development of halteres in Drosophila. The third thoracic segment of the fly typically bears halteres, small balancing organs crucial for flight stability. The Hox gene ultrabathorax, UBX, is the key regulator in this process. UBX represses the expression of wing genes in the third thoracic segment, preventing the development of a full-fledged wing and leading to the formation of halteres instead. However, mutations in UBX can disrupt this repression, resulting in the dramatic transformation of halteres into a second pair of wings, creating the iconic four-winged fly. This dramatic phenotype vividly illustrates the power of UBX in controlling segment identity and appendage development. The combinatorial nature of the Hox code adds another layer of complexity to this regulatory landscape. Different combinations of Hox genes can specify a wide array of structures, generating the remarkable diversity observed across animal body plans. In the developing limb of a mouse, for example, the specific combination of Hox genes expressed determines the identity of the different limb segments: the stylopod, upper arm; zeugopod, forearm; and autopod, hand. The precise boundaries and levels of expression of HOXA9 to HOXA13 and HOXT9 to HOXT13 dictate the formation of the humerus, radius, ulna, carpals, metacarpals, and phalanges. This combinatorial code allows for a fine-grained control of limb development, ensuring the correct formation of each limb segment. The development of the first cervical vertebra in mice provides another compelling example of the combinatorial Hox code at work. HOXA3 and HOXT3, two paralogous genes, are both expressed in the anterior cervical vertebrae, near where the first neck vertebra articulates with the skull. While a mutation in HOXA3 alone has no discernible effect, a mutation in HOXT3 causes a partial fusion of the first neck vertebra with the skull. However, a double mutation, affecting both HOXA3 and HOXT3, results in a complete fusion of the vertebra with the skull. This observation demonstrates that HOXA3, although seemingly redundant, plays a crucial role in the proper development of the first cervical vertebra and highlights the importance of considering the combined effects of multiple Hox genes. The story of Hox genes is not confined to embryonic development. Their influence extends into later stages, particularly in insects undergoing metamorphosis. In larvae with gradual metamorphosis, Hox genes are activated in larval tissues, primarily in the trunk region, that will persist into adulthood. This ensures the continuity of the body plan throughout the larval and adult stages. However, in larvae with complete metamorphosis, Hox genes are expressed predominantly in the juvenile rudiments, the precursors of adult structures, while being absent in the transient larval tissues. This reflects the fundamental difference in developmental strategies between these two types of metamorphosis. The consequences of Hox gene misregulation can be far-reaching, leading to a range of developmental defects. In Drosophila, mutations in the antennapedia (ANTP) gene can cause the transformation of antennae into legs, a dramatic homeotic transformation that illustrates the gene's crucial role in specifying antennal identity. In humans, altered Hox coding can lead to the development of Barrett's esophagus, a condition where the lining of the esophagus is replaced by tissue resembling the lining of the intestine. This metaplasia, a change in cell type, is a precursor to esophageal cancer, highlighting the link between Hox gene misregulation and oncogenesis. Hox gene mutations can also disrupt the delicate balance of Hox gene regulation, leading to skeletal abnormalities in vertebrates. Mutations in HOX5, HOX6, HOX9, HOX10, or HOX11 genes in mice can cause transformations of specific vertebrae, altering their shapes and characteristics. These transformations underscore the crucial role of Hox genes in specifying the identity of vertebral elements along the 8p axis. The staggered domains of Hox gene expression along the body axis are essential for the proper development of the vertebral column, and disruptions to this pattern can have profound consequences for skeletal morphology. The remarkable conservation of Hox gene function is further demonstrated by cross-species experiments. The ability of a fly to function relatively normally with a chicken Hox protein replacing its own highlights the deep similarities in their molecular mechanisms, despite their evolutionary divergence. This functional conservation reflects the ancient origin and fundamental role of Hox genes in shaping animal body plans. The story of Hox genes is a story of molecular addresses, combinatorial codes, and evolutionary ingenuity. These genetic architects, with their precise molecular tools and intricate regulatory networks, orchestrate the development of complex structures, ensuring that the right parts form in the right places at the right times. Their influence spans the animal kingdom, uniting us in a shared genetic heritage. As we continue to unravel the secrets of Hox genes, we gain a deeper appreciation for the remarkable mechanisms that have shaped the diversity of life on Earth. Chapter 4. The Hox Paradox: Redundancy, Robustness, and the Mystery of Missing Genes. The Hox genes, as we've seen, are master architects of the animal body plan. They direct the formation of everything from the delicate mouthparts of a fly to the complex vertebrae of our own spines. But as we delve deeper into the world of Hox genes, we encounter a fascinating paradox, a blend of redundancy, robustness, and the surprising absence of these seemingly essential genes in some organisms. This chapter explores these seemingly contradictory themes, revealing the flexibility and adaptability of developmental programs. One of the most intriguing aspects of Hox genes in vertebrates is their redundancy. Unlike the simpler system often found in insects, where each segment typically relies on a single Hox gene for its identity, vertebrates often have multiple Hox genes with overlapping roles. This redundancy might seem wasteful. Why have multiple genes doing the same job? The answer, it turns out, lies in the concept of robustness. This built-in backup system ensures that development can proceed smoothly even if one gene is lost or damaged. Consider the development of the first cervical vertebra in mice. Two Hox genes, HOXA3 and HOXD3, are expressed in this region. Deleting HOXA3 alone has no noticeable effect. The vertebra develops normally. This might lead one to believe that HOXA3 is unimportant, a genetic spare part. However, deleting both HOXA3 and HOXD3 reveals a different story. The vertebra doesn't form correctly, demonstrating HOXA3's hidden importance. HOXD3 can compensate for the loss of HOXA3, but if both are missing, development goes awry. This redundancy acts as a safety net, ensuring developmental stability in the face of genetic perturbations. This principle of redundancy extends beyond the HOXA3-D3 example. In mice, the four Hox clusters, HOXA, HOXB, HOXC, and HOXD, often contain paralogous genes, genes with similar sequences and overlapping expression patterns. This paralogy contributes to the robustness of the Hox code, the combinatorial expression of Hox genes that defines segment identity. For example, in the development of the mouse axial skeleton, different combinations of Hox genes specify the different types of vertebrae: cervical, thoracic, lumbar, sacral. If one gene in the combination is lost, others can often compensate, maintaining the correct vertebral identity. This combinatorial Hox code in vertebrates contrasts sharply with the simpler, one gene per segment system often found in insects. In Drosophila, for example, the identity of the second thoracic segment, where the wings develop, is largely determined by the antennapedia gene. A mutation in this single gene can dramatically transform the antennae into legs. Similarly, the ultrabathorax gene specifies the identity of the third thoracic segment, where halteres develop. A mutation in ultrabathorax can transform the halteres into a second pair of wings. This one-to-one relationship between Hox genes and segment identity in insects makes them highly susceptible to dramatic homeotic transformations caused by single gene mutations. The difference in Hox gene organization and function between vertebrates and insects has important implications for evolutionary flexibility. The redundant, combinatorial code of vertebrates allows for more subtle variations in morphology. Changes in the expression levels of one Hox gene can be buffered by other genes in the combination, leading to gradual evolutionary changes. In contrast, the simpler Hox code of insects, while more susceptible to dramatic transformations, can also lead to rapid evolutionary innovations. The evolution of diverse wing patterns in butterflies, for example, may be linked to changes in the regulation of Hox genes expressed in the developing wings. The Hox paradox deepens when we consider organisms that seem to thrive despite missing Hox genes that are considered essential in other species. The hemichordate Schizocardium californicum and the Palladium larva of Nemertea, for instance, do not express Hox genes in their larval stages. Hemichordates are marine invertebrates, closely related to chordates, with a body plan that includes a proboscis, collar, and trunk. Nemerteans, also known as ribbon worms, are another group of marine invertebrates with a simple, unsegmented body plan. The absence of Hox gene expression in the larval stages of these organisms challenges our understanding of their absolute necessity for body patterning. Even more surprising is the case of the miniature fishes of the genus Petacyprus. These tiny fish, among the smallest vertebrates in the world, exhibit an extreme form of developmental truncation, meaning they reach sexual maturity while retaining many larval characteristics. This developmental shortcut is accompanied by a significant loss of Hox genes. At least 7 Hox genes, normally present in related fish species like zebrafish, are missing in Petacyprus. This loss of Hox genes mirrors the truncated development of these fish, suggesting a link between Hox gene number and developmental complexity. The loss of Hox genes in Petacyprus may be an adaptation to their extreme environment, the acidic blackwaters of Southeast Asian peat swamps. The reduced genome size, resulting from the loss of Hox genes and other genetic material, may offer a selective advantage in this resource-limited habitat. This example highlights the surprising ways organisms can adapt and thrive even with seemingly incomplete Hox gene sets. Other examples of Hox gene loss or variation can be found throughout the animal kingdom. Some fish species have undergone additional Hox cluster duplications, resulting in 7 or more clusters. This expansion of Hox genes may have contributed to the remarkable diversity of fish body plans. In contrast, some invertebrates, like nematodes, have experienced Hox gene loss, with their Hox gene set being smaller and less clustered than in other animals. These variations in Hox gene number and organization reflect the diverse evolutionary pressures acting on different lineages. The cases of Schizocardium californicum, the Palladium larva of Nemertea, and Petacyprus highlight the remarkable plasticity of developmental programs. These organisms, despite lacking certain Hox genes, have evolved successful body plans and life histories. Their existence challenges the notion of a universal Hox code and underscores the importance of considering the interplay between Hox genes and other developmental factors in shaping morphology. The absence of Hox genes in some organisms doesn't necessarily mean a lack of body patterning. It simply means that other mechanisms have evolved to fulfill this role. The Hox paradox, with its seemingly contradictory themes of redundancy, robustness, and missing genes, reveals the remarkable adaptability of developmental programs. The Hox code, while providing a fundamental framework for body patterning, is not a rigid set of rules. Organisms have evolved diverse strategies for utilizing and modifying this code, adapting to a wide range of environments and life histories. The story of Hox genes is a story of both constraint and opportunity, of ancient conservation and evolutionary innovation. As we continue to explore this fascinating genetic landscape, we gain a deeper appreciation for the intricate mechanisms that have shaped the diversity of life on Earth. Chapter 5. When Hox Goes Wrong: Homeotic Transformations and the Price of Misregulation. The Hox genes, as we've explored, are the master architects of the animal body plan, orchestrating the intricate dance of development with remarkable precision. But what happens when these meticulous directors make a mistake? When Hox genes go awry, the consequences can be dramatic, revealing their crucial role in ensuring the correct development of body structures. Mutations or misexpression of these genes can lead to homeotic transformations, where one body part is replaced by another, sometimes with startling results. One of the most iconic examples of a Hox gene mutation is the antennapedia mutation in fruit flies. In these mutants, legs sprout from the head where antennae should be, a dramatic shift in appendage identity. This transformation is caused by a dominant mutation in the antennapedia gene, which normally specifies the development of legs in the second thoracic segment. The mutation causes the gene to be misexpressed in the head, leading to the inappropriate development of legs in this region. Another striking example from Drosophila involves the ultrabathorax (UBX) gene. This gene normally represses wing development in the third thoracic segment, allowing the formation of halteres, small balancing organs. Mutations in UBX can disrupt this repression, leading to the development of a second pair of wings, creating the four-winged fly. This dramatic phenotype highlights UB's crucial role in specifying segment identity. But Hox gene misregulation is not limited to dramatic transformations of appendages. Mutations in other Drosophila Hox genes can lead to a range of developmental errors. For instance, mutations in labial can disrupt head involution, a critical process where the fly's head structures are internalized during embryonic development. This can result in missing or malformed mouthparts and salivary glands. Similarly, mutations in deformed can cause head defects or even transform head segments into thoracic segments. These examples underscore the importance of precise Hox gene regulation for proper head development. Hox gene mutations can also affect the development of other body structures. Mutations in proboscipedia, another Drosophila Hox gene, can disrupt the formation of mouthparts, transforming them into leg-like structures. This illustrates the role of proboscipedia in specifying the identity of the mouthparts. Moving beyond Drosophila, we find examples of Hox gene misregulation in other arthropods. Variations in the expression of Hox genes 7, 8, and 9 can shift the position of maxillopeds, specialized appendages involved in feeding, in crustaceans and other arthropods. These shifts can alter the number of segments associated with maxillopeds, demonstrating the role of Hox genes in coordinating segmental identities. Hox genes also play a critical role in specifying the body regions of trilobites, extinct marine arthropods. Fossil evidence suggests that trilobites had three body regions: the cephalon, head, thorax, and pygidium, tail, each characterized by a unique combination of Hox genes. Changes in Hox gene expression during trilobite evolution likely contributed to the diversification of their body plans. In vertebrates, Hox gene mutations can lead to skeletal abnormalities. Studies in mice have revealed that mutations in Hox5, Hox6, Hox9, Hox10, or Hox11 can cause transformations of specific vertebrae, altering their shapes and characteristics. For example, mutations in Hox10 genes can transform lumbar vertebrae into thoracic vertebrae, highlighting the role of these genes in specifying the identity of different vertebral types. Misexpression of Hox genes, both spatially and temporally, can also disrupt development. The ultrabathorax gene in Drosophila provides a compelling example. While loss-of-function mutations in UBX can lead to four-winged flies, misexpression of UBX in the second thoracic segment, where wings normally develop, can have the opposite effect, transforming wings into halteres, resulting in a four-haltered fly. This transformation involves the repression of wing genes by UBX, including genes like blistered, which is involved in cell adhesion in the wing blade, and spalt, which patterns the placement of wing veins. The timing of Hox gene expression is also crucial for proper development. In insects with gradual metamorphosis, Hox genes are expressed in larval tissues that will be carried over into adulthood, ensuring the continuity of the body plan. However, in insects with complete metamorphosis, Hox genes are primarily active in the juvenile rudiments, the precursors of adult structures, and are largely absent in the transient larval tissues. This difference in timing reflects the distinct developmental strategies of these two insect groups. The connection between Hox gene misregulation and human health issues extends beyond limb malformations. Hox genes play a role in the development of several types of cancer and leukemia. Altered Hox coding can disrupt cellular differentiation and contribute to oncogenesis, the process by which normal cells transform into cancer cells. For example, misexpression of HOXA9 is associated with acute myeloid leukemia, a type of blood cancer. Similarly, mutations in HOXA13 and HOXD13 are linked to hand-foot genital syndrome, a condition characterized by limb malformations and urogenital abnormalities. The involvement of Hox genes in human diseases is often complex, with redundancy and compensatory mechanisms playing a role. The HOXA3, HOXD3 example in mice illustrates this complexity. While a mutation in either gene alone may not cause severe defects, a double mutation can have profound consequences. This redundancy can make it challenging to pinpoint the precise role of individual Hox genes in human diseases. The study of Hox genes in miniature fishes like Pedicipris has revealed further insights into the link between Hox genes and development. These tiny fish exhibit an extreme form of developmental truncation, reaching sexual maturity while retaining larval characteristics. This truncated development is accompanied by a significant loss of Hox genes, suggesting a connection between Hox gene number and developmental complexity. The loss of these genes in Pedicipris may be an adaptation to their extreme environment. The MLL gene, the human homologue of the Drosophila gene trithorax, plays a critical role in regulating Hox gene expression. Chromosomal translocations involving the MLL gene can create fusion genes that disrupt Hox gene regulation and contribute to the development of aggressive leukemias. These fusion genes often result in the overexpression or misexpression of Hox genes, leading to uncontrolled cell proliferation and cancer development. The stories of Hox gene misregulation, from the Antennapedia mutation in flies to the complex involvement of Hox genes in human cancers, highlight their profound influence on development and health. These examples underscore the importance of precise Hox gene regulation for normal development and the dramatic consequences when these genetic architects go astray. The ongoing research into Hox genes continues to unravel their intricate roles in shaping animal form and function, offering valuable insights into the fundamental processes of life and the development of human diseases. Chapter 6. Hox genes gone rogue, when master architects turn malignant. We've journeyed through the intricate world of Hox genes, marveling at their role as master architects of the animal body plan. These genetic conductors orchestrate the developmental symphony, ensuring the right structures emerge in the right places at the right times. But what happens when these conductors go rogue, their harmonious melodies turning into a cacophony of misregulation? This chapter explores the darker side of Hox genes, revealing their unexpected involvement in diseases like cancer and leukemia, where their very power over cellular identity becomes a weapon turned against the body. Hox genes, despite their primary role in embryonic development, don't simply retire after birth. They continue to be expressed throughout our lives, playing essential roles in tissue maintenance and repair. They're like diligent construction crews, constantly ensuring the structural integrity of our bodies. But sometimes these normally meticulous workers become corrupted, their precise instructions scrambled, leading to uncontrolled growth and the development of cancer. One way Hox genes can go rogue is through misexpression. Imagine a building where the blueprints for the foundation are accidentally used for the roof. The resulting structure would be unstable, prone to collapse. Similarly, when Hox genes are expressed in the wrong tissues or at the wrong times, they can disrupt the normal balance of cellular processes, leading to uncontrolled cell growth and the formation of tumors. For instance, HOXA9, a Hox gene normally involved in limb development, is often overexpressed in acute myeloid leukemia, a type of blood cancer. This overexpression disrupts the normal differentiation of blood cells, leading to an accumulation of immature, cancerous cells. Another Hox gene, HOXB13, normally involved in the development of the urogenital system, plays a sinister role in prostate cancer. Overexpression of HOXB13 in prostate cells promotes uncontrolled growth and contributes to the development of tumors. This highlights how a Hox gene's normal function in development can be hijacked by cancer cells to fuel their uncontrolled proliferation. The connection between Hox genes and cancer becomes even more apparent when we consider the concept of metaplasia. This is a process where one cell type transforms into another, like a construction worker suddenly deciding to become a plumber, disrupting the carefully planned division of labor. Hox genes, with their influence over cell fate, can be key players in this transformation. For example, in Barrett's esophagus, a condition that can lead to esophageal cancer, the cells lining the esophagus change into cells resembling those found in the intestine. This metaplasia is associated with altered Hox gene expression, suggesting that these genes play a role in the cellular transformation that predisposes individuals to cancer. The story of Hox genes in cancer also involves chromosomal translocations, a phenomenon where segments of DNA swap places between different chromosomes. These translocations can disrupt the delicate regulatory landscape of Hox genes, leading to their misregulation. One notorious example involves the MLL gene, the human counterpart of the Drosophila gene trithorax. MLL normally plays a crucial role in regulating Hox gene expression, ensuring their proper function during development. However, translocations involving MLL can create fusion genes, hybrid genes that combine parts of MLL with parts of other genes, often including Hox genes themselves. These MLL fusion genes act like saboteurs, disrupting the normal function of Hox genes and other developmental pathways. They can lead to the overexpression or misexpression of Hox genes, driving uncontrolled cell growth and contributing to the development of aggressive leukemias. The story of MLL translocations highlights the devastating consequences of disrupting the delicate balance of Hox gene regulation. Hox genes are not just involved in the development of specific tissues, they also play a crucial role in establishing positional identity, the cellular equivalent of a GPS system that tells cells where they are in the body. This positional information is critical for the proper organization and function of tissues and organs. During limb development, for example, Hox genes along the AP axis specify the identity of different limb segments. The Hox A and Hox D clusters, in particular, are crucial for patterning the limbs. Genes like Hox A9 to 13 and Hox D9 to 13 determine whether a cell will contribute to the upper arm, forearm, or hand. This positional information is also critical for preventing metastasis, the spread of cancer cells from their original site to other parts of the body. When Hox genes are misregulated in cancer, cells can lose their positional identity, becoming disoriented and migrating to inappropriate locations. Imagine construction workers, their GPS systems malfunctioning, building a section of wall in the middle of a hallway instead of its proper place. Similarly, cancer cells that have lost their positional identity can break away from the primary tumor and travel to distant sites, establishing new tumors and spreading the disease. The link between Hox genes and cancer is further reinforced by the concept of oncology recapitulating ontology. This phrase captures the idea that cancer development often mirrors the processes of embryonic development. Hox genes, so crucial in orchestrating the formation of tissues and organs during embryogenesis, can be reactivated or misregulated in cancer cells, driving their uncontrolled growth and the formation of tumor-like structures. It's as if the body's developmental program is hijacked by cancer cells, using the very tools that built the body to fuel its own destruction. The study of Hox genes in hand-foot genital syndrome provides another example of how their misregulation can lead to developmental defects. Mutations in HOXA13 and HOXD13, two Hox genes involved in limb development, can cause hand and foot malformations, as well as urogenital abnormalities. These mutations disrupt the normal patterning of the limbs, leading to shortened or fused digits and other skeletal abnormalities. The involvement of Hox genes in a wide range of cancers, from leukemias to solid tumors, underscores their pervasive influence on cellular identity and behavior. Their misregulation can disrupt the delicate balance of cellular processes, leading to uncontrolled growth, metaplasia, and metastasis. The concept of oncology recapitulating ontology highlights the deep connection between development and disease, reminding us that the same genes that build our bodies can also contribute to their demise. As we continue to unravel the complex roles of Hox genes in cancer, we gain valuable insights into the molecular mechanisms of disease progression, opening new avenues for developing targeted therapies and improving patient outcomes. The study of Hox genes, these powerful architects of life, continues to reveal both the wonders of normal development and the devastating consequences when their control is lost. Hox genes also play crucial roles in the development of other organ systems, such as the hindbrain in vertebrates. The hindbrain is the posterior part of the brain, responsible for vital functions like breathing, heart rate, and balance. Hox genes are expressed in specific segments of the hindbrain called rhombomeres, and their precise expression patterns are essential for the proper development of these segments. Mutations in Hox genes can lead to defects in hindbrain segmentation, resulting in neurological disorders. The story of Hox genes is not just about their roles in normal development and disease. It also encompasses the evolution of these genes and their regulatory networks. Hox genes are ancient and highly conserved, meaning they have remained relatively unchanged over vast evolutionary timescales. Their presence in diverse animal groups, from flies to humans, suggests that they played a fundamental role in the evolution of animal body plans. The organization of Hox genes into clusters is also an ancient and conserved feature. However, the number and arrangement of Hox clusters can vary between different animal groups. Insects typically have a single Hox cluster, while vertebrates have 4. This expansion of Hox clusters in vertebrates may have contributed to the increased complexity of their body plans. The regulation of Hox genes is also subject to evolutionary change. The cis-regulatory elements, the DNA sequences that control Hox gene expression, can evolve rapidly, leading to changes in Hox gene expression patterns and the evolution of new morphological features. For example, the evolution of different wing patterns in butterflies may be linked to changes in the cis-regulatory elements of Hox genes expressed in the developing wings. The study of Hox genes in diverse animal models, from flies to fish to mice, has provided invaluable insights into their function in evolution. These studies have revealed the remarkable conservation of Hox gene function across vast evolutionary distances, as well as the surprising diversity of their roles in different organisms. The ongoing research on Hox genes continues to expand our understanding of the fundamental principles of development, evolution, and disease. These genetic architects, with their ancient origins and powerful influence, continue to shape the story of life on Earth. Chapter 7. Rewriting the Code, Exploring the Frontiers of Hox Gene Manipulation. Our exploration of Hox genes has revealed their profound influence, shaping the animal kingdom's diverse forms, from the intricate body plan of a fruit fly to the complex anatomy of a human. But our understanding of these master architects is not merely an academic pursuit. Scientists are actively seeking ways to harness the power of Hox genes, to rewrite the developmental code itself, for therapeutic purposes. This chapter delves into the cutting edge of Hox gene manipulation, exploring its potential to revolutionize regenerative medicine and cancer treatment, while acknowledging the inherent challenges and ethical considerations. One of the most promising tools in this endeavor is CRISPR-Cas9, a revolutionary gene editing technology that allows scientists to make precise changes to DNA sequences. Imagine a surgeon with molecular scalpels, able to target and correct individual genetic errors. CRISPR-Cas9 offers this level of precision, enabling researchers to correct mutations in Hox genes that cause developmental disorders. For instance, scientists have used CRISPR-Cas9 to correct Hox gene mutations in human cells grown in the lab, paving the way for potential therapies for congenital abnormalities. The zebrafish, a small, transparent fish, has proven invaluable in Hox gene research. Its transparent embryos allow scientists to observe developmental processes in real time, making it an ideal model for studying Hox gene function. Researchers have used CRISPR-Cas9 to knock out specific Hox genes in zebrafish, revealing their roles in fin development. Knocking out HOXD13, for example, leads to shorter fins with fewer bony rays, highlighting the gene's role in promoting fin outgrowth. These studies not only illuminate the function of individual Hox genes but also inform potential therapeutic strategies for limb malformations in humans. Beyond correcting mutations, researchers are exploring ways to manipulate Hox gene expression to regenerate damaged tissues. Hox genes, with their ability to control cell fate, hold the key to unlocking the body's regenerative potential. Studies in mice have shown that increasing the expression of HOXA10, a gene normally active in the developing limbs, can revert skeletal progenitor cells to a more primitive, self-renewing state. These rejuvenated cells can then be used to repair bone fractures, offering a potential therapy for age-related bone loss and other skeletal disorders. The potential of Hox gene manipulation extends beyond bone regeneration. Researchers are investigating its application in regenerating other tissues, such as cartilage, muscle, and even spinal cord. Manipulating Hox genes could stimulate the growth of new, healthy tissue, offering hope for patients with injuries or degenerative diseases. Imagine a future where spinal cord injuries are no longer irreversible, where damaged hearts can be repaired, and where lost limbs can be regrown. Hox gene manipulation holds the promise of making these regenerative dreams a reality. The connection between Hox genes and cancer offers another avenue for therapeutic intervention. As discussed in the previous chapter, Hox genes can go rogue, their misregulation contributing to the development of various cancers. By understanding how Hox genes contribute to cancer development, researchers can devise strategies to target these genes and disrupt the cancerous process. For example, inhibiting the overexpression of HOXA9 in acute myeloid leukemia could potentially halt the uncontrolled proliferation of cancerous blood cells. Similarly, targeting HOXB13 in prostate cancer could slow tumor growth and prevent metastasis. The concept of a Hox code, the combinatorial expression of Hox genes that specifies cell fate, is central to these therapeutic strategies. By deciphering this code, researchers can learn how to manipulate Hox genes to direct specific cell fates for therapeutic benefit. For instance, by manipulating the combination of Hox genes expressed in stem cells, scientists could potentially guide their differentiation into specific cell types needed for tissue repair or regeneration. Imagine a future where we can reprogram skin cells into heart cells or nerve cells, providing a readily available source of cells for treating a wide range of diseases. The intricate interactions and regulation of Hox genes add another layer of complexity to these therapeutic endeavors. Hox genes don't act in isolation. They interact with each other and with other genes in complex regulatory networks. These interactions must be carefully considered when designing therapeutic strategies. Manipulating one Hox gene could have unintended consequences on the expression of other genes, potentially disrupting developmental processes or even causing harmful side effects. The power of Hox genes comes with significant ethical considerations, especially when considering their manipulation in humans. These genes are fundamental to our development, shaping our very form and identity. Altering Hox genes could have unforeseen and potentially irreversible consequences. The ethical implications of manipulating human development are profound, raising questions about the boundaries of scientific intervention and the potential for unintended consequences. Careful consideration and open discussion are crucial to ensure that these powerful tools are used responsibly and ethically. The therapeutic frontier of Hox gene manipulation is a landscape of both immense promise and daunting challenges. The ability to rewrite the developmental code offers unprecedented opportunities for treating a wide range of diseases. However, the complexity of Hox gene regulation and the ethical considerations surrounding their manipulation necessitate a cautious and thoughtful approach. As we continue to explore the frontiers of Hox gene research, we must navigate this landscape with both scientific rigor and ethical responsibility, ensuring that these powerful tools are used to improve human health and well-being while upholding the highest ethical standards. The development of Barrett's esophagus provides a compelling example of how Hox gene misregulation can contribute to disease. In this condition, the cells lining the esophagus, normally a squamous epithelium, transform into a columnar epithelium resembling that of the intestine. This metaplasia, a change in cell type, is a precursor to esophageal cancer. Studies have shown that altered Hox gene expression is associated with Barrett's esophagus, suggesting that these genes play a role in the cellular transformation. By understanding the precise role of Hox genes in this process, researchers could develop new diagnostic tools or targeted therapies to prevent or reverse the metaplasia and reduce the risk of esophageal cancer. The remarkable functional conservation of Hox genes across vast evolutionary distances offers unique opportunities for cross-species research. The fact that a fly can function with a chicken Hox protein substituting for its own highlights the deep similarities in their molecular mechanisms. This conservation allows researchers to use simpler model organisms, like Drosophila and zebrafish, to study Hox gene function and regulation, gaining insights that can be translated to more complex organisms, including humans. The zebrafish, with its transparent embryos and well-established genetic tools, is particularly well suited for studying Hox genes in real time during development. By manipulating Hox genes in zebrafish, researchers can gain a deeper understanding of their roles in limb development, fin regeneration, and other developmental processes, informing potential therapeutic strategies for human diseases. The miniature fishes of the genus Petacypris, with their extreme developmental truncation and associated loss of Hox genes, offer a unique perspective on the relationship between Hox genes and developmental complexity. These tiny fish reach sexual maturity while retaining many larval characteristics, a phenomenon linked to the absence of at least 7 Hox genes normally found in related fish species. Studying these fish could provide valuable insights into the minimal Hox gene set required for vertebrate development and the evolutionary pressures that can lead to Hox gene loss. The study of Hox genes has also revealed their involvement in human limb malformations. Mutations in HOXA13 and HOXD13, 2 Hox genes involved in limb development, can cause hand-foot genital syndrome, a condition characterized by limb and urogenital abnormalities. Understanding the precise molecular mechanisms by which these mutations disrupt limb development can inform the development of new diagnostic and therapeutic approaches for this and other congenital limb malformations. The story of Hox genes is a testament to their enduring importance in shaping the diversity of animal life. From their roles in specifying segment identity in flies to their involvement in human diseases, these master architects continue to fascinate and inspire researchers. As we continue to unravel their secrets, we gain a deeper appreciation for the intricate mechanisms that govern development and evolution, opening new avenues for therapeutic intervention and a deeper understanding of the very fabric of life. Chapter 8. The Future of Form, Hox Genes and the Promise of Regenerative Medicine. Our exploration of Hox genes has revealed their profound influence on shaping the animal kingdom, orchestrating the intricate development of everything from a fly's delicate mouthparts to the complex vertebrae of our spines. But our understanding of these master architects is not merely an academic pursuit. It holds the potential to revolutionize medicine, offering a glimpse into a future where damaged tissues regenerate, spinal cords are repaired, and even lost limbs might be regrown. This chapter delves into the promise of Hox genes in regenerative medicine, exploring the exciting possibilities while acknowledging the significant challenges that lie ahead. The very nature of Hox genes, their inherent role in directing the formation of body structures during embryonic development, makes them prime candidates for regenerative therapies. Imagine our bodies as intricate buildings, meticulously constructed according to detailed blueprints. Hox genes are the architects of these blueprints, directing the placement of walls, windows, and doors, the tissues, organs, and limbs that make up our bodies. If a building is damaged, we consult the original blueprints to guide the repair process. Similarly, scientists envision using Hox genes to reactivate the body's developmental programs, guiding the regeneration of damaged tissues. One of the most tantalizing prospects is the regeneration of the spinal cord. Spinal cord injuries, often resulting in paralysis, are notoriously difficult to treat. The complex architecture of the spinal cord, with its intricate network of neurons and supporting cells, makes repair a formidable challenge. However, Hox genes offer a potential solution. These genes play a crucial role in patterning the spinal cord during development, specifying the identity and position of different types of neurons. By manipulating Hox gene expression, scientists could potentially stimulate the growth of new neurons and guide their integration into the existing circuitry, restoring lost function. Another area of intense interest is heart tissue regeneration. Heart attacks, a leading cause of death worldwide, often leave behind damaged heart muscle that struggles to pump blood effectively. Hox genes, with their influence over cell fate, could be used to reprogram cells into heart muscle cells, replacing the damaged tissue and restoring heart function. Studies have shown that manipulating Hox gene expression can revert cells to a more primitive, stem cell-like state. These rejuvenated cells could then be directed to differentiate into heart muscle cells, providing a source of new, healthy tissue for heart repair. The most audacious goal of Hox gene manipulation in regenerative medicine is the regeneration of lost limbs. While limb regeneration occurs naturally in some animals, like salamanders, it is a far more limited process in humans. Hox genes, with their crucial role in limb development, hold the key to unlocking this regenerative potential. By manipulating Hox gene expression in the remaining limb tissue, scientists could potentially stimulate the growth of a new limb, complete with all its intricate structures, bones, muscles, nerves, and blood vessels. One promising avenue for achieving these regenerative goals is the use of induced pluripotent stem cells, iPSCs. These cells, derived from adult tissues like skin, can be reprogrammed to an embryonic stem cell-like state, capable of differentiating into any cell type in the body. By manipulating Hox gene expression in iPSCs, scientists could generate specific cell types needed for tissue repair, such as neurons for spinal cord regeneration or heart muscle cells for heart repair. This approach offers the potential to create patient-specific therapies, minimizing the risk of rejection. The research on bone regeneration in mice provides a compelling example of the therapeutic potential of Hox gene manipulation. Studies have shown that increasing the expression of HOXA10, a Hox gene normally active in the developing limbs, can revert skeletal progenitor cells to a more primitive, self-renewing state. These rejuvenated cells can then be used to repair bone fractures, offering a potential therapy for age-related bone loss and other skeletal disorders. This research suggests that Hox genes could be used to rejuvenate aging tissues, restoring their regenerative capacity. However, the road to harnessing the regenerative power of Hox genes is fraught with challenges. These genes don't act in isolation. They interact with each other and with other genes in complex regulatory networks. Manipulating one Hox gene can have unintended consequences on the expression of other genes, potentially disrupting developmental processes or causing harmful side effects. The intricate interplay between Hox genes and their cofactors, proteins that modulate their activity, adds another layer of complexity. Understanding these interactions is crucial for developing safe and effective Hox gene therapies. The anatomical location also plays a critical role in Hox gene function. Hox genes exhibit spatial collinearity, meaning their expression along the body's AP-axis corresponds to their position within the Hox cluster. This spatial specificity must be considered when designing Hox gene therapies. For instance, HOXA10 is preferentially expressed in the tibia, while HOXA5 is preferentially expressed in the ribs. Manipulating HOXA10 might be effective for regenerating tibia bone, but not necessarily for regenerating rib bone. Similarly, manipulating HOXA5 might be more effective for regenerating rib bone. This anatomical specificity requires a targeted approach to Hox gene therapy, tailoring the treatment to the specific tissue or organ being targeted. The tools for manipulating Hox genes are constantly evolving. CRISPR-Cas9, with its ability to make precise changes to DNA sequences, offers unprecedented precision for correcting Hox gene mutations. Other gene editing technologies, such as zinc finger nucleases and talons, also hold promise for manipulating Hox genes. In addition to gene editing, researchers are exploring techniques to modulate Hox gene expression using small molecules or RNA interference. These approaches could offer less invasive ways to manipulate Hox gene activity for therapeutic benefit. The ethical implications of Hox gene manipulation, especially in humans, are profound. These genes are fundamental to our development, shaping our very form and identity. Altering Hox genes could have unforeseen and potentially irreversible consequences. Careful consideration and open discussion are essential to ensure that these powerful tools are used responsibly and ethically, with the utmost respect for human dignity and well-being. The future of regenerative medicine is undeniably intertwined with our understanding of Hox genes. These master architects hold the key to unlocking the body's regenerative potential, offering hope for treating a wide range of diseases and injuries. While significant challenges remain, the ongoing research into Hox gene manipulation provides a beacon of hope for a future where our bodies can heal and regenerate in ways we can only dream of today. As we continue to explore the intricate world of Hox genes, we move closer to realizing the full promise of regenerative medicine, a future where the body's own blueprint can be harnessed to rebuild and restore. Hox genes are the master architects of the animal kingdom, orchestrating the intricate dance of development that shapes our bodies and those of countless other creatures. From the humble fruit fly to ourselves, these remarkable genes dictate the formation of body structures, ensuring that the right parts emerge in the right places at the right times. This exploration has revealed their profound influence, from their ancient origins and evolutionary conservation to their complex regulation and the devastating consequences of their misregulation. We've learned that Hox genes operate through a precise spatiotemporal code, a carefully choreographed symphony of gene expression that determines the identity of different body regions. This code, while remarkably conserved across diverse species, also allows for the incredible variety of animal forms we see today. We've seen how mutations in Hox genes can lead to dramatic transformations, such as legs growing where antennae should be in flies, highlighting their crucial role in normal development. We've also explored the darker side of Hox genes, their involvement in diseases like cancer and leukemia, where their very power over cellular identity can be turned against the body. The implications of this research extend far beyond the laboratory. Understanding Hox genes provides fundamental insights into the processes that shape life itself, from the earliest stages of embryonic development to the maintenance and repair of tissues throughout our lives. This knowledge has the potential to revolutionize medicine, paving the way for regenerative therapies that could repair damaged tissues, regenerate spinal cords, and even regrow lost limbs. The ability to manipulate Hox genes offers a glimpse into a future where we can harness the body's own blueprint to rebuild and restore. The journey of Hox gene research is far from over. Scientists continue to unravel the intricate regulatory networks that govern Hox gene expression, exploring the complex interplay between these genes and their cofactors, as well as the influence of environmental factors. New technologies, like CRISPR-Cas9, provide powerful tools for manipulating Hox genes with unprecedented precision, opening exciting new avenues for therapeutic intervention. However, the power of these tools comes with significant ethical considerations, requiring careful thought and open discussion about the responsible use of these technologies. What are your thoughts on the potential of Hox gene manipulation? What ethical considerations do you think are most important? Share your thoughts and questions in the comments below. We encourage you to like and subscribe for more explorations into the fascinating world of science. And remember, the pursuit of knowledge is a journey without end, fueled by our innate curiosity and the desire to understand the world around us. Never stop asking questions, and never stop learning.